physician scientist at and the Richard M. Isabel Furlaud Professor and head of the Laboratory of Chemical Biology and Signal Transduction at the Rockefeller University in New York. His research interests are quite expansive, actually, but it's focused on how cells communicate with each other and the environment. If you're going to be studying cell signaling or outside to inside cell signaling or signal transduction, you're likely to come across the importance of G protein coupled receptors. So here, Tom has really made incredible, important discoveries on the fundamental GPCR biology and has advanced our understanding of GPCRs as a druggable therapeutic target. Tom got his bachelor's in chemistry and his medical degree from the University of Chicago and went on to do his residency at Massachusetts General Hospital in Boston and did a postdoc with Gobind Khurana at MIT. And it was here that he really embraced the powers of molecular biology and what I like to call Legos for adults, being one of the first to perform site-directed mutagenesis on GPCRs and heterologous expression. I can say from hearing him speak a couple times that he has not shied away from the powers of mutagenesis, and I'm sure he'll be talking about that today. He is recruited to Rockefeller University in 1990, where he has been ever since. He has pioneered studies in rhodopsin and understanding the chemical basis for spectral tuning and color vision. He has studied chemokine receptors and their importance in cell movement and HIV entry into the cell. In using GCE and bioorthogonal chemistry, he has studied membrane receptor dynamics and signaling. There's no real like a way I can go through all the accomplishments of Tom, so I'll just highlight some here. That he's trained over 45 postdoctoral fellows, 15 graduate students, and 70 summer scholars. And looking through the list, many of them have gone on to be leaders in both academia, biotechnology, and pharmaceutical industries. Tom has published over 200 papers as a Howard Hughes medical investigator. And in 2020, he was awarded an honorary doctorate from the Karolinska Institute, which is, I don't know how you do that, but that's awesome. Honorary degree. So today he'll be telling us about applications of genetic code expansion and bioorthogonal chemistry to study membrane proteins. Thanks, Tom.
Thanks, John. Please just confirm that you can hear me and see the screen okay. Are we good?
Everything's good.
So first of all, thanks to the organizers, thanks to John, Rick, thanks to Andy for reaching out to me about this program. I'm really excited to be able to give a talk today about our latest work that uses genetic code expansion. And what we're really trying to do in this particular project, or actually program, is to use genetic code expansion to create receptor biosensors that we can use as a drug discovery technology. So I'm going to actually start with acknowledgments in case I run over time. But this is my lab at Rockefeller University in New York City. And let's see, I'll use the laser pointer. The work that I'm going to talk about was, although it's a team effort, as you'll see, including with colleagues from Pfizer, the team leader was Jordan Mathiasen, who's right here. She got her PhD this past year at Rockefeller, working in my lab, and is now a postdoctoral fellow at AstraZeneca in Boston. And Jordan here got a lot of attention for some of the work that I'm going to describe to you today, which is published. But Jordan also did something really amazing during the pandemic when the swimming pools locally were closed down. She took up open water swimming to keep up her swimming career, and ended up 2 summers ago swimming around Manhattan. There's a program called the 20 Bridges Tour, which is 28.5 miles of open water swim. It's a sanctioned event. And on one beautiful July day, she started at the tip of Manhattan, swam up the East River, right by the lab, and we were all cheering for her, and made it to the tip in Upper Harlem, made a left turn, and then swam down the Hudson River back to the starting point. So that amazing accomplishment. I mean, I've met a lot of people in science who've done a lot of amazing things, but I've never met a scientist who swam around Manhattan before. So Jordan, congratulations on that and the work I'm going to talk about. So just a brief introduction to GPCR biology. GPCRs are 7 transmembrane helical receptors. They reside primarily in the plasma membrane, although there's a lot of interesting cell trafficking going on with GPCRs. A GPCR is activated by an endogenous agonist. That agonist could be a small molecule, a metabolite, a lipid, a small protein, a peptide, depends on the receptor. And we'll see in a minute, there are hundreds of these receptors in typical cells. By definition, they interact with a heterotrimeric G protein, and really the function of the receptor in its active state is to induce the release of GDP by the heterotrimeric G protein. That release of GDP activates the receptor. Activates the heterotrimeric G protein. GTP is uptaken by the G protein, which then dissociates into alpha and beta gamma subunits. And those subunits interact with cellular effectors like adenylyl cyclase or phospholipases and really induce second messenger level changes in the cell, which drive the signaling event. The turnoff of the receptor is complicated, but involves phosphorylation and binding of arrestin. But the arrestin pathway, which is independent from the G protein pathway generally, is really interesting in and of itself and relates to the turnoff of the signal. Now, the interesting thing, if you look at this subunit, it's really the receptor plus heterotrimeric G protein in the membrane is the signaling complex or the signalosome or whatever you want to call it. But it's an allosteric machine. The reason is that agonist ligand binding in the orthosteric binding pocket of the receptor induces a conformational change, which is transmitted about 60 to 80 angstroms across the membrane into another protein and to the nucleotide binding pocket of the alpha subunit to release the GDP. So this complex of receptor plus heterotrimeric G protein is an allosteric machine, and the activation of the receptor and its turnoff can be affected allosterically, and we're gonna talk about that in a second. Well, let's see if I can figure out how to advance the slide. GPCRs are important drug targets, and about 30% of all FDA-approved drugs actually target GPCRs. GPCRs are the largest receptor family in the human genome, about 800 members, about 300 or so of them are actually ligand-activated receptors that are potential drug targets. Drugs for GPCRs are increasingly important and but remain challenging. And if you look at the importance of GPCR drugs and how they're distributed across these 400 or so receptors, you see that most drugs are antagonists, and many drugs target the same receptor. So you have multiple drugs that target beta receptors or dopamine receptors, for example. There are a few agonists that are available, but in general, you have the whole phylogenetic tree that is not targeted. Less than 10% of GPCRs actually have been drugged so far. And there's a lot of interesting work that remains to be done in this system. This inset shows maybe part of the reason is that 90% of all the work on GPCRs focuses on about 25 receptors. And those receptors happen to be the ones that are drugged, or maybe they happen to be the ones drugged because most of the work has been done on them for a variety of reasons. They are tractable, they're easy to study, they're easy to express. Whatever the reason, there are hundreds of GPCRs that are not studied almost at all. And this is one of the reasons that work is challenging. Most of you have heard about recently Ozempic and Wegovy and Modar. I forget the third name of the third one, but these obesity and diabetes treatments, these are GPCR targeted. They target GLP-1 and GIP receptors, which are in the secretin family of receptors, absolute blockbuster drugs. And these drugs sort of came out of nowhere recently, although there were peptide analogues that had been out there for a while. So there's increasing interest in GPCR drug discovery. Now, if we look at where drugs are located in structures of the receptor, you'll see that basically the drugs occupy orthosteric binding sites in the receptors. The orthosteric binding site is by definition the site in three-dimensional space where the endogenous ligand, which is almost always an agonist, binds in the receptor. Now, it turns out that it's challenging to target orthosteric sites because all of the receptors share the same site, and it's difficult often to get specificity of those reactions because you have a limit in this size of this orthosteric site. However, the new frontier, I think, is in allosteric modulation. What is an allosteric modulator of a GPCR? Well, first of all, the allosteric site for a potential allosteric drug is a site, a binding pocket on the receptor that is distinct from the orthosteric site. So it's distinct from the orthosteric site, but what does it do pharmacologically? Normally, drugs are either antagonists for the agonist or so-called inverse agonists, which means that they suppress any basal activity that the receptor might have even when the agonist is not present. So typical drugs are inverse agonists or antagonists. In a few cases, like for the case of Ozempic, the drugs are agonists that have particular pharmacological properties that can be manipulated by making, for example, an analog to the normal agonist, that's the endogenous agonist. But allosteric drugs would bind in other pockets on the receptor. And this is possible because the receptors are allosteric machines. They undergo conformational changes that are ligand dependent. And those changes can be modulated by activity at second sites. Now, there are two kinds of allosteric modulators, so-called positive allosteric modulators or negative allosteric modulators. And positive means basically enhanced signaling, negative decreased signaling. But these definitions are really based on kinetic principles. A positive allosteric modulator would typically decrease the off rate for a bound agonist, and a negative allosteric modulator would decrease the would increase the off rate for the agonist. So we're talking about binding at second sites from the orthosteric site that affect the activity through its effect on the binding of the agonist itself. Now, this is kind of an attractive drug property, if you think about it, because you can still have the normal physiological function at the receptor target, but you're just modulating that function, fine-tuning it, for example, with a drug entity. The problem is, although people have realized what I just said for maybe at least a decade, so far there are only 2 or maybe 3 allosteric drugs of the hundreds of drugs targeting GPCRs. The problem is that typical drug screening paradigms, including cell-based screens, are not very good at finding drugs that bind at allosteric sites. It's not always easy to figure out what the functional effect might be of an allosteric modulator. It's difficult to define kinetic properties in cell-based screens, which are usually pseudo-steady state kind of equilibrium assays. And often the allosteric site would have a lower ligand affinity than the orthosteric site. So what is the best way to look for orthosteric drugs for given targets? What we would like to do is develop biosensors using genetic code expansion. Now, there are some tool compounds available to help us with this, but none of these compounds, as shown here for so-called class A receptors, there are about 10 different allosteric modulators for class B and class C. And you see in the overlay of a typical structure of a class A receptor, these bind in a location different than the orthosteric site, which is up here. Some of these on this surface actually overlap with the orthosteric site, but there are other allosteric small molecules that bind totally on the other side of the receptor. Same thing for family B, and this is even more pronounced, where you have orthosteric binding sites for these tool compounds in the transmembrane domain or outside of the helical bundle, but within the membrane embedded region of the receptor on the opposite side of the receptor from where the agonist binds, which is here. Class C, which are the metabotropic glutamate receptors and other receptors like that, also have a number of allosteric modulators. Class C, remember, has a big N-terminal domain, extracellular domain here, which is not shown in this particular figure. So how can we set up a screen for molecules like this in a cellular environment where we don't know a priori what we're looking for? So traditionally, a binding first approach was taken for allosteric discovery, but it's not always clear which sites might be allosteric. And in fact, sometimes allosteric sites, since they target the active state of the receptor, are cryptic sites when the receptor is in its off state or inactive state. Those sites become apparent only upon receptor activation. And of course, it's becoming easier to find such sites with the advent of cryo-EM structural biology, where a receptor can be imaged in its active state in complex with a heterotrimeric G protein, for example, in a nanodisk or in some other kind of membrane mimetic environment. So more insights are coming from structural biology, but it's slow going still. And also, low affinity interactions are not always well suited for cell-based screens. And a lot of the work in this area has originated from in vitro biophysical studies, where you can actually purify large amounts of material and look at drug binding interactions directly. But GPCRs, of course, are difficult to purify in high amounts that are suitable for biophysical studies. So we wanted to try a function first approach. Could we identify allosteric sites of interest? Can we figure out how to increase the affinity of test compounds for these sites in live cell systems? And could, in fact, we accomplish these aims by creating engineered biosensors for cell-based screens? And that's what I'm going to talk about today. All of this work is in our ongoing program to use genetic code expansion to study GPCRs. And we've, in fact, used genetic code expansion for about 15 years now, going back to Xixin Yi in 2008, We have applied IR probes to study rhodopsin activation. We figured out how to do targeted photo-crosslinking, similar to what Isabella described for channels. We've been doing that in GPCRs for 10 or 12 years. And we've figured out how to apply bioorthogonal coupling chemistries to link fluorophores site-specifically into GPCRs, for example. And now I'll tell you about this last portion of this program, Jordan's work on engineering biosensors. I talked a little bit about this concept of tethering drug fragments. And the tethering concept is really coming from the work of Jim Wells and others that led to this so-called fragment-based drug screening. I won't go into detail about that, but the proof of concept work on fragment-based screening was done on protein targets that could be purified and studied in biophysical assays, in which a cysteine was present near the active site, and drug fragments could be linked into this active site using sulfhydryl chemistry. In that formulation, a drug fragment can be tethered to the active site, which allows screening of smaller libraries of low affinity binders into this site. And if there's a hit, then you have essentially the possibility to link the original fragment with the new fragment that you discover by screening in medicinal chemistry after the fact. And then you have a protein targeted with this fragment-based approach. The problem is that this isn't really amenable. This cysteine-based approach isn't really amenable for studies of GPCRs. So we wanted to try to see if we could install a different reactive residue near a site of interest besides a cysteine. And this is sort of the workflow or the experimental plan that we started with. If we had an allosteric site and a target receptor, could we install a non-canonical amino acid that had a functional group near to the site, but without affecting the function of the receptor? That could be monitored in a cell-based system. Could we then design a drug fragment that could be linked covalently using a bioorthogonal reaction in a cell-based system to that non-canonical amino acid? In that case, we might get some binding of the fragment without tethering, or we might get some tethering of the fragment without binding, but most likely we would get binding and tethering, or at least some equilibrium between all of these possibilities. And we could be able to distinguish among these using a cell-based model. So all of this is going to happen in live cells that are amenable for screening. Everything that we did in this project was done in HEK cells, and the assay we're going to use is a calcium mobilization assay, where we look at the effect of an agonist ligand on these receptors, plus minus the tether. And you'll see how that's done in a dose response. But before we did that, we tried to make sure it would work. We did math modeling of this reaction. I won't go into the details, but this is described in the papers. We had a toolkit that we've developed earlier for the fluorescence labeling, where we use two different systems, the tyrosine RS or the pyrrolysine RS, with three different chemistries. The SPAC reaction, the reaction that's well known in Oregon State, the inverse electron demand Diels-Alder reaction, which we really love, and three different amino acids, azido-Phe, CPK, TCOK. And so basically everything that I'm going to tell you today was done with all three of these amino acids, but the one that worked best and the one I'll focus on is the CPK amino acid with a tetrazine or methyltetrazine drug fragment derivative with the Diels-Alder reaction to get this kind of a project. Now, to screen these different approaches, We developed a luminescence assay that we could do in microtiter plate format. Basically, it's fairly straightforward. We just link luciferase to the C-terminal tail of our receptor of interest. We introduce the amber stop codon, do the genetic code expansion reaction in the presence of the non-canonical amino acid. If we get incorporation and read-through, then we get luciferase activity, and we can correlate luciferase activity plus minus amino acid at different sites in the protein with 3 different systems here, the azido-Phe, the CPK, and a TCOK. So this is a great screen for us, and we spent a lot of time figuring out what was best. And it turned out that what was best was the CPK and the Diels-Alder reaction. We used CCR5 as our model system. It's a chemokine receptor, and there is an allosteric modulator of CCR5. This drug is commercially marketed as an HIV co-receptor blocker, so it's given to HIV/AIDS patients in conjunction with other HIV therapies. And this drug is marketed by Pfizer. So early on in this project, we went to Pfizer hoping that they would have a lot of medicinal chemistry in the freezer based on their Moraviroc drug discovery program, and hoping that they might want to work with us on this project because the receptor's already been targeted. So obviously we have to be able to publish this. It's a proof of concept, and we would be working on a receptor that they've already targeted, not something that's a secret that they're planning to target. So we looked at the structure of Moraviroc bound to CCR5. We picked a couple of sites for the potential to introduce the non-canonical amino acid without affecting activity. We showed that we could do that in these dose response curves with the agonist, which is called RANTES, and the inhibition of the agonist-induced calcium flux with the drug Moraviroc. So basically, if we look at this CPK case, at these different positions, the positions here are essentially similar to wild type. This 184 in particular looks pretty good. And a lot of the work I'll show is at that 184 position. So our friends at Pfizer, with our input, synthesized analogues of all of these of Moraviroc with different linkers with either tetrazine or methyltetrazine on the backbone of the BCNs, the DBCOs, and the CPKs, which are not shown here, not to mention the azido compounds that are also aren't shown here. So we had a whole library of tetherable drug fragments. And this is just one example of how we would do this. If we just take Moraviroc, basically Moraviroc with a linker with methyltetrazine, we can see that if we react it, first of all, the wild type and the 184 CPK are indistinguishable. If we add the analog M6, we get some inhibition, but it's not the same as the Moraviroc case. With the CPK and the analog, however, we get a left shift, showing that the ability to couple this group to the CPK increases the affinity of this tool compound, this analog of Moraviroc, by almost 2 orders of magnitude. Now that's kind of interesting and led to the acceleration of this program with a lot of different analogs, showing, for example, that some work and some don't, depending on factors such as the linker length, the type of fragment we have, and the type of chemistry we're using. But the goal is to find a low affinity fragment that we can then tether near the allosteric site to get the observed high affinity effect. And that's what we show here, for example, with this M12 compound, which is this compound. It's still a fragment of Maraviroc, but it doesn't have any binding affinity at all in the wild type receptor. However, if we add it to the receptor that has a CPK at position 184, we get a nice dose response curve. It's not as good as Maraviroc, but it's definitely measurable in our cell-based system. Whereas without the tether, there's no affinity at all, no binding at all. So we can take this information and do, for example, a pulse chase experiment with M12 to show that we actually get tetherable binding, covalent binding of that fragment that's dependent on the fragment also having an affinity for the binding pocket. So it's a interesting give and take, which was predicted by the math model. You get tethering, which increases the affinity for the binding pocket, but you don't get tethering unless you have some affinity for the binding pocket. And this is very cool. We can do a pulse chase with a fluorophore that's linked to the methyl-tetrazine with the same spacer, for example. Here's the TET 680 that we use, and we're following up all in a cell-based system, the dose response curves with the fluorescent dose response experiment. And of course, there's another way we can reduce the affinity of the drug fragment other than make chemical analogues of Maraviroc. The other way is to put the tether at locations that aren't near to the site that we're trying to probe. And we chose 184, for example, because it was known to be close to the binding site. But we can do, in principle, any other mutation anywhere in the protein. It doesn't affect expression. And you can see then that we can look at reactivity of the low affinity compound versus functionality. And the reactivity and functionality are coordinated. I'm not going to show all the details, but we can essentially use this information to model the location of the allosteric binding pocket to the particular residue that we're putting the CPK at, and then optimize this proof of concept experiment. Here would be the Maraviroc, here's the CPK, and we can then measure distances between the CPK and the Maraviroc using MD simulations. And we can also, on the same receptor construct, do the dose response curve showing the function and the pulse chase fluorescence experiment. So all of this is basically done on one construct, some in silico, some calculations, and some in situ in the cell-based system. And of course, we can do this for all of these mutants and all of the analogs. This is just one example of one particular analog. And we see that there's really a very cool correlation between the distance constraints and the functional observations, which leads us to believe that this might actually work for a drug screen. So again, this was our approach. We showed that we could actually do this and link drug fragments that are amenable to drug-based screening. And we did this using genetic code expansion that you all are aware of, but we were able to devise a nice screening system to compare directly in the same cells with the same receptor target, different chemistries to do the bioorthogonal labeling reactions. And then everything after that we did in the live cells, just like we normally do a drug screen. So we're basically just adding a step upstream in a normal type of a fragment-based screen. And the real nice aspect of the proof of concept is that we could correlate efficiency of coupling with the location of the non-canonical amino acid that we're introducing, which means that this is amenable to rational design of the cell-based biosensor constructs that we're engineering. I think this is significant because we really need a better approach to discover allosteric drug candidates for GPCRs. And this, in principle, is very cool because it's generalizable to any GPCR target. It's probably more useful if there's a crystal structure of that target, but really not required, especially in the advent of AlphaFold and really advanced AI-based homology modeling. So I end where I began, thanks to Jordan, especially, who worked with Tori and Emily, and Thomas Huber, of course, did a lot of the computational work and has been a great colleague in the lab over the years of this entire project. So thanks very much. And if there are any questions, I'd be happy to take them. Thanks for your attention and for sticking around.
That was great. Thanks, Tom, for the great talk. And again, if there's any questions, you can put it in the chat, the Q&A. I'll start off the question. So that was really interesting. It's kind of like a molecular tape measure for GPCRs, kind of similar to maybe like what Chris Miller did with potassium channels with TEA block back in the day, which is really cool, I think. Have you thought about doing photoisomerization of the linker to put it in and out of where it would bind and maybe make a kind of a GPCR or druggable by light and look at kinetics there?
Yeah, I really love these photo on systems, switch back and forth between on and off. And we're trying to figure out how we might do that. And it's doable in this system because now we have so much information about the proper linker length, what the tethered fragments actually look like in the binding pocket from MD simulations. So it wouldn't be outrageous to add sort of this photo-on technology, which is really cool. We haven't used it, but we want to use it. So thanks for the, maybe I'll give you a call later to talk about how we would do that.
Yeah, it's really neat. Is there any other questions from anybody? Yeah. Looks like one of the attendees has a question. They raised their hand. Can we make them allowed to talk?
I could just ask my question here.
Oh, here you are, Ryan. It's you.
So, Tom, a beautiful work again. Always, I love the pioneering aspect to hit really hard problems with GCE. I really. So one quick question on my side was, you explored a number of different chemical reactivities, obviously, to identify sort of what was the right tethering chemistry. But what I need is just a brief understanding. I didn't quite catch why, like, each of those different chemistries have sort of different reaction rates, different reaction extents. What was the sort of silver bullet about that?
Yeah. So the silver bullet is we want to be able to do the assay in the same cells that we do the chemistry in, and we want to do that in a microtiter plate format. So the reaction rate and efficiency of the reaction in terms of generally a low expressing membrane protein target was the key. And it was so important that we even spent a lot of time with the reaction that looked like it would be the best, seeing if there were differences between, for example, tetrazine and methyltetrazine in the in your reaction. And it turns out there are differences, and some of the differences have to do not with the reaction rate itself, but with the stability of the methyltetrazine versus tetrazine. Methyltetrazine just seemed to be a little bit more stable for longer periods of time when we added it to the cell culture media. So a lot of what we did was, we understood the underlying principles of the chemistry, but a lot of it was trial and error to figure it out. And that's why Jordan spent so much time comparing these different chemistries. It was, I must say, it was time consuming, but we're really happy with the performance of CPK in particular and your chemistry with the methyltetrazine especially. So
No, I truly appreciate the difficulty of all of that optimization. I know so much of that sometimes gets buried in those beautiful papers, right? And it was just, it's good to hear sort of how much, I've always believed that it has to essentially be optimized inside a cell. And it's great to understand that basically that's really just what you had to go through for your particular system. Totally makes sense. Thank you much.
You're welcome. Thank you.
Are there any other questions? If not, we're running over a little bit on the time, but I'd like to thank the speakers again, Isabella and Tom, for 2 great talks. Thank you. And they will be available if you go to the GCE for All website, if you want to watch them later. Thanks again, everybody. Have a great day or night.
Thanks, John.
Thank you.
Thanks, everyone.
Thanks, Tom. That was great.